Hello students, I am Dr. Divya from TeenEinstein. Now we are starting the second chapter, Sexual Reproduction in Flowering Plants. In 12th standard biology, the first part that we are learning is reproduction, right? So we have already completed our first chapter, reproduction in organism. So reproduction means that is a vital process, and you know very well that without reproduction a species cannot survive. Okay, there won't be continuity for the species. So each individual that leaves its progeny by either asexual reproduction or by sexual method. So there will be either asexual reproduction or sexual reproduction in all organisms. They will be following any one of this method. So asexual reproduction means that makes exact copies of that organism. Asexual reproduction makes exact copies of that organism, and they are called as clones. Otherwise they are called as clones. Asexual reproduction will give rise to clones, means there won't be any difference between the parental organism and the newly formed organism. Okay, so there won't be any difference between those organisms. In first generation and the second generation there won't be any difference. So asexual reproduction makes clones, but sexual reproduction makes new varieties or new type of individuals, new hybrids. So in our first chapter, the reproduction in organism, we have learned about asexual reproduction and sexual reproduction, isn't it? Now in our second chapter, it is a continuation of the first chapter what we have learned about asexual and sexual reproduction. So here we are going to continue, or in this chapter we are going to learn the procedures or the events of that reproduction of sexual reproduction. So reproduction, it's of two types, asexual and sexual. We have learned already asexual reproduction and sexual reproduction. So in this, asexual reproduction means there is no fusion of gametes. There is no gametic fusion in the case of asexual reproduction. So in this asexual reproduction that we can see in algae, fungi and all. In algae, fungi and all, we can see asexual reproduction that we have learned in the first chapter. Means mainly we can see asexual reproduction in prokaryotes, and unicellular organism also adopt the same asexual method of reproduction, or asexual mode of reproduction can be seen in unicellular organism or prokaryotes. But the sexual reproduction, in sexual reproduction what takes place? Fusion of gametes, that is there, fusion of gametes. Gametic fusion is taking place in sexual reproduction. So sexual reproduction means that is the fusion of male and female gametes, that is called as sexual reproduction. So sexual reproduction is it's not at all a simple process. It's a very complex process, and it's very slow process because so many events are there in a sexual reproduction. Sexual reproduction means it's a complex process and slower process, and compared to asexual reproduction, it's a complex process. Asexual reproduction means exact copies are making. The organism is going to make exact copies in asexual reproduction. But in the case of sexual reproduction, it is a complex process and it is a time consuming or slower process. Compared to asexual reproduction, sexual reproduction is a slower and complex process. Why it is called as a slower process? Because so many events are taking place, or so many steps are involved in the sexual reproduction. And most of the higher organisms, they reproduce by sexual mode. So sexual reproduction is seen in higher organism. Mainly it is seen in higher organism. So sexual reproduction is taking place in different steps. Sexual reproduction is taking place in different steps. They are called as pre-fertilization, then fertilization, then post-fertilization. Pre-fertilization means prior to fertilization, what and all, or the organism is getting ready. And fertilization means that is the process of fertilization or the fusion of gametes. Then post-fertilization means after fertilization, what and all taking place. So sexual reproduction is categorized into several events. They are pre-fertilization, fertilization and post-fertilization. Okay. So in pre-fertilization events, the plant itself, we are discussing about the sexual reproduction of plants. So in pre-fertilization, plant is getting ready, or that includes gametogenesis or the formation of male and female gametes, and gametic transfer, the transfer of this formed male and female gametes. For what? For fusion. Okay. So pre-fertilization means the plant is getting ready for the fertilization. How the plant will get ready? It will form the gametes, then it will transfer the gametes to a particular place for the fusion. Got it? So in the fertilization, which is the second step, or after pre-fertilization, fertilization is coming. So in fertilization, already the plant made everything ready, means the gametes are ready, male and female gametes are ready. Now in fertilization, these formed male and female gametes will fuse together, or it is called as syngamy. Syngamy is the fusion of male and female gametes. So these gametes will fuse together in the process of, or in the procedure of fertilization. It is nothing but the fusion of male and female gametes. Then after fertilization, next is the events which are coming after fertilization, that is called as post-fertilization events. So the fused gametes that will form a zygote, and these zygote will undergo division, and this division, the continued division, will form an embryo. Embryo is formed after a continuous division of the zygote. So this is called as embryogenesis. Embryogenesis is nothing but the formation of embryo. Embryogenesis is called as, it is coming under post-fertilization events. The formation of embryo, it is called as embryogenesis. Got it? So up to this we have learned in the first chapter, right? So all these things we have learned in first chapter. So in this chapter, the second chapter, we are going to explain all these procedures in a very common way. So we are talking about reproduction in organism. That's what we have discussed in the first chapter. In the first chapter, we explained everything like the pre-fertilization events and the post-fertilization events and fertilization. About all this in a common way we have explained. We have given an explanation in a general way. But in the second chapter, we are going to learn the sexual reproduction in flowering plants. So we are going to learn each and every step in detail in this chapter. But we are going to learn about plants, sexual reproduction in flowering plants. Okay? So the flower sexual reproduction in flowering plant. In this chapter, we are going to learn about flower and its structure. Okay, and we have already learned about different steps of sexual reproduction. So in this chapter, we are going to learn different steps of sexual reproduction in flowering plants. Okay, that means about pre-fertilization. We are learning about pre-fertilization. Then we will learn the structures which are involved in this pre-fertilization and the events involved in this. Then the same way we will learn about fertilization steps and post-fertilization structures and events also. So we will learn one by one. So the main thing is we are going to learn the structure of a flower. So the flower. Let's move to the topic, the flower. So now we are learning about sexual reproduction of flowering plants, right? So all flowering plants that shows sexual reproduction. We know that almost all flowering plants shows sexual reproduction, and the sexual reproduction you can see in angiosperms. So the flowering plants, they are also called as angiosperms, right? We have learned about plant kingdom, and in that angiosperms are the advanced level of plants or the higher plants, right? They are the higher plants. Angiosperm means with hidden seeds. They have the hidden seeds and they produce seeds. Angiosperms means with hidden seeds and they produce seeds. Angiosperms, they have lot of diversity, or they are diverse group of plants. And in gymnosperms, the seeds are exposed. What is the difference between angiosperms and gymnosperms? Angiosperms have covered seeds. In the same case, gymnosperms have naked seeds, or their seeds are exposed. The word meaning is exposed seeds. Gymno means naked seeds. They have the naked seeds in the case of gymnosperms. But in angiosperms, the seeds are found in the flower. So just imagine the common flower like marigold. We can see seeds. Where we can see the seeds, it is inside the flower, right? So in angiosperms, you can easily make out the male and female reproductive organs also, right? So all flowering plants, they show sexual reproduction, right? The flowering plants, they are also called as angiosperms. They are called as angiosperms, means plants with hidden seeds. So they are the most diverse group of land plants. Which are the most diverse group? Angiosperms. They shows different types of plants. Okay, the seeds of angiosperms, they are found in the flower. Where the seeds are present? Inside the flower. So, but in the case of gymnosperms, the seeds are exposed. In gymnosperms, seeds are born exposed to the bodies and surface of the reproductive part. Then in angiosperms, both male and female organs can easily be found in the flowers of angiosperms. Both male and female organs we can easily make out in the case of angiosperms. In the flower, both male and female organs are, it's very easy to identify the male and female organs in the case of angiosperms. So in the flower, both male and female organs will be present. Okay, here you can see a common flower, hibiscus. So almost every part we are going to explain with the help of this flower only, hibiscus. Is it clear? The next, we said angiosperms are reproducing by sexual method, right? So in sexual reproduction, what are all the events included in sexual reproduction? In this, both parents, that means the male and female parent, they are involved in the reproduction. Both male and female parents are involved. In each parent they are producing sex cells, right? Each parent is producing sex cell, and these sex cells, they are called as gametes, right? So in the case of sexual reproduction, both male and female parents, both type of parents are involved, and each parent, that means the both the male and female parent, both parents are producing sex cells, and these sex cells are called as gametes. So the male parent is producing male gamete, and the female parent is producing female gamete. Okay. The male parent is producing male sex cell or male gamete, and the female parent is producing female sex cell or the female gamete. Next, the next step is the male and female gamete. Both are formed now. Then next, what is going to happen? The male and female gamete, they are fusing. There is a fusion of male and female gamete. So in the case of angiosperms, flowers, they are the sites of sexual reproduction. So all these events like pre-fertilization, post-fertilization and fertilization, everything is taking place inside the flower. Got it? So all these things or all these events are happening inside the flower. So what are all the features of sexual reproduction? In this, and what is in sexual reproduction, mainly two parents are involved and they are producing a new individual. Without the involvement of male and female parents, there is no sexual reproduction. So in sexual reproduction, two parents are involved and they are producing a new individual. So this offspring or the new individual produced by the fusion of gametes or sex cells, it is called as sexual reproduction. So flowers are the sites of sexual reproduction in angiosperms. In angiosperms, flowers are the sites of sexual reproduction. And the features of sexual reproduction are, in sexual reproduction, two parents are involved. They are the male parents and female parents. Both male and female parents are involved in sexual reproduction. And the male parent will produce the male gamete and the female parent will produce the female gamete. So these gamete formation and fertilization, that is another feature of sexual reproduction. There is a gamete formation, male parent will produce or that will form male gamete and female parent will produce female gamete. So the gamete formation is another feature, and the fertilization, that is another feature of sexual reproduction. There is a gamete formation, one step, and there is a fertilization. Fertilization is nothing but the fusion of this male and female gamete. So that is the feature of sexual reproduction. Then this whole process, it is slow and lengthy. It is a complex, very complex process, and it is time consuming process. In sexual reproduction, there is formation of male and female gamete and fertilization is taking place. The whole process is very lengthy and it is a slow process. Okay, so the variation occurs. What do you mean by variation is there in the offspring? That offsprings, they are formed with the help of a male and female gametic fusion, right? That offspring is produced by gametic fusion. So they'll be having the male parent character and female parent character also. So both these characters getting fused and it is forming a new individual. So offsprings are different. They are showing difference from the parents. Not like the asexual reproduction. Asexual reproduction is producing exact clones or exact characters. Like the parental organism, the same features will be present in the newly formed or the offspring also. But in the case of sexual reproduction, there is a fusion of both male and female gametes. So the offsprings are different from the parents. Genetically also they are different, and physically also they will show the difference. Genetically as well as physically, these parents and offsprings are different. Got it? Next, we said flower is the site of sexual reproduction in angiosperms. right? Flower is the site, or in flower all these pre-fertilization, fertilization and post-fertilization events are taking place inside the flower in the case of angiosperms. So we should know the structure of a flower. First we should learn the structure of a flower, then only we can learn how these procedures or the events are taking place inside a flower, right? So we should know the structure of a flower, or how the flower is making arrangements for this pre-fertilization, fertilization and post-fertilization events. How these events are taking place inside a flower. We should learn the structure of a flower to know these events, or to learn about these events. So in your textbook they have given flower, a fascinating organ of angiosperms. Here I am using the same title for this, for presenting this topic, because flower, it is really a fascinating organ of angiosperms, because a small flower, inside a very small flower also all these events are taking place. So it's really amazing, or it is really wonderful. So a beautiful flower, think about a beautiful small flower. Inside that also all these events are taking place. This pre-fertilization, fertilization, then post-fertilization. All these events are taking place in a minute flower also. Right? So the title says flower is a fascinating organ of angiosperms. So exactly it is a fascinating organ only, because everything is happening inside the flower, from the small seed, and after all this fertilization procedure it is forming a small seed, and from that small seed a very big plant or a tree is arising, right? But that seed formation, it is taking place inside a flower, right? As a result of this pre-fertilization, fertilization and post-fertilization events, inside the flower a seed is getting produced. So the flower is not a simple organ, right? It is really a fascinating or a wonderful organ of higher plants, or angiosperms. And it's not a simple process. I said it's a complex and time consuming, or a lengthy process or procedure. So the flower is the reproductive part of higher plants. Both male and female gametes, they are produced by the flower. The flower is producing both the male and female gamete, and in the flower the sexual reproduction is taking place. Where sexual reproduction is taking place? Inside the flower. So that is the site of sexual reproduction in angiosperms, right? So let's learn the structure of a flower, a complete flower. After learning the complete flower, or the structure of a complete flower, we will learn the function of each part. So first, a complete flower is having 4 parts. Which are the 4 parts? Calyx, corolla, androecium and gynoecium. These are the 4 main parts of a complete flower. A complete flower will have calyx, corolla, androecium and gynoecium. What is calyx? Calyx consists of sepals. These are the sepals. You can see here the green part. Here you can see. Those are called as sepals. Calyx is sepals. Next is corolla. Corolla means petals. Corolla is nothing but petals. Petals, the colorful part. And androecium. Androecium is the male organ. Androecium. Androecium is male organ. Here you can see the androecium. And gynoecium is the female part. Gynoecium is the female part. You can see this is the gynoecium. This is the female part. So sepal, petal, anther and stigma or style. All these things, these are the essential: calyx, corolla, androecium and gynoecium. So in a flower, here you can see these are the sepals. Sepals. Calyx means calyx consists of sepals. Next is corolla. Corolla means the colorful part, the petals. Then androecium. Androecium consists of stamen. Here you can see stamen. Androecium consists of stamen, that is a male part. Then gynoecium. Gynoecium consists of carpel. This structure, this is the carpel. So a carpel, it is having stigma, style and ovary. Okay. So in these calyx and corolla, they are the non-essential whorls or the non-essential parts of a flower. If calyx, that means sepal, and corolla means petal, these both are not there also, a flower can do sexual reproduction. Then androecium and gynoecium, these are called as the essential whorls or essential parts of a flower. Why it is the essential, it is becoming the essential parts? Because without androecium, androecium is the male reproductive organ and gynoecium is the female reproductive organ. Without these, sexual reproduction is impossible. So they are called as the essential whorls or essential parts of a flower. Okay. So the flower, it is the reproductive part of a plant. Both male and female gametes, they are produced by a flower. And the sexual reproduction in plant, it is taking place inside the flower. Even it is a minute flower or big flower, sexual reproduction is taking place inside the flower. So the complete flower, a complete flower is having 4 parts: calyx, corolla, androecium and gynoecium. Calyx means sepal. Corolla means petals, and this calyx and corolla, they are the non-essential whorls of a flower. Next is androecium. Androecium consists of stamen and gynoecium consists of carpel. This androecium and gynoecium, they are the essential whorls of a flower. Without androecium and gynoecium, sexual reproduction is impossible, or it's not possible, it won't take place. So these are the essential whorls of a flower. So before flowering, the plant will go through different stages, right? So pre-fertilization, we are going to learn about the pre-fertilization structures and events. Before fertilization, the things, what and all happening in the plant, that's what we are learning now. So in pre-fertilization, before flower formation itself, a plant will go through different stages, right? So before flowering or before the actual flower is seen on the plant, that plant will decide that it is going to flower, or the plant, it will get ready for flowering. So according to that, all the changes, it will take place in the plant, like hormonal changes will happen and structural changes also occurs. And when it is ready for flowering, or when it is perfect for a flowering period, the bud formation will start, and that bud will grow and it will form the flower. So that time, the formation of a floral primordium or the floral bud and differentiation of that floral primordium, it is taking place. Got? Before flowering, the plant itself will plan, or it will decide that it is going to flower. So according to that, the changes which are happening in a plant, according to that, what and all changes will happen, hormonal changes will happen, structural changes will happen. So all these things will start, and it will, these events will happen one by one. Okay, one after the other. First the plant will grow, then some kind of changes will happen, hormonal changes, internal structural changes, then bud formation will start, then this bud will grow and it will form a complete flower. Then the plant is going for this fertilization and such procedures, and after fertilization and seed formation, the plant will die. It will undergo senescence or death. There is a senescence phase also in the plants. So the plant will go through different stages. So there are 3 stages in the life cycle of an angiosperm. First stage is the vegetative stage. In vegetative stage, only growth is taking place. That is a growing period of a plant. And next stage is the reproductive stage. Here you can see the vegetative stage, that is growing stage of a plant. That is nothing but the growing stage. And second stage is the reproductive stage. Reproductive stage means a plant is getting ready for a reproduction. And the third stage is the senescence stage. Senescence stage means the plant is becoming old and it is near to the final stage, or it is near to death. And when a plant is changing from vegetative phase to reproductive phase, and before flowering, there are number of structural and hormonal changes. There are a number of structural as well as hormonal changes in the plant when it is changing from vegetative to reproductive stage. Right? This is a growing stage. From that it is reaching or it is attaining a mature stage. Then next stage is the reproductive stage. So from the vegetative stage to reproductive stage, it will, when it is changing, the plant is changing, it will undergo lot of changes like structural as well as hormonal changes. Got it? And in this picture, you can see apical meristem. That apical meristem of this shoot, it is transforming to reproductive meristem. Here you can see the apical meristem is transforming to reproductive meristem. All these things are occurring due to hormonal changes. A plant is growing, that time some kind of hormones will be present, then it is in the vegetative stage, it will grow, then another hormonal changes and structural changes are taking place inside the flower, inside the plant, that due to that flower formation will start, flowers will start, and after flowering it is ready for fertilization, then pollination is taking place, fertilization is taking place, and fruit formation. That is also a kind of structural as well as hormonal change. So there are number of changes, hormonal as well as structural changes. Then this reproductive meristem, that will grow and slowly that forms the inflorescence. You can see here, this is the growing stage. Then that reproductive meristem will grow and it will slowly form the inflorescence, or flower formation will take place. What is inflorescence? The arrangement of a flower on a stem. This kind of arrangement of a flower on a stem. Okay. On an axis, the flowers are getting arranged. Okay. So first this inflorescence, that axis develops. If that, in the case of inflorescence, first that axis develops, and afterwards on that inflorescence axis, floral primordia or floral buds will develop. First floral primordia or floral buds will arise. So first from this down, that floral buds are arising, and these at the tip, the last formed floral buds will be present. Then this floral primordia, that will grow into floral buds. You can see it will grow into floral bud, and then it will form the complete flower. Right? So shoot apical meristem is transformed into reproductive meristem. It is transformed into reproductive meristem. You can see here this shoot, that apical bud, it will grow and it will form a reproductive meristem. That reproductive meristem grows to form inflorescence axis. It will form inflorescence axis, and on that inflorescence axis, floral primordia will develop, and these floral primordia grows into floral buds. It will form floral buds, and later it will form flowers. So in a flower, we have already discussed, in a flower there are 4 parts: calyx, corolla, androecium and gynoecium. These are the 4 floral parts of a flower: androecium, gynoecium, calyx and corolla. So in this calyx and corolla, we have already told they are the non-essential parts, because it is not present, then also fertilization will take place, right? Without calyx and corolla also fertilization is possible. And for reproduction we need androecium and gynoecium. So these both are called as essential whorls, essential parts or essential whorls of a flower, because these are the male and female structures. Without that, the sexual reproduction, it is impossible. So in a flower, the androecium and gynoecium both are developing, right? Both are developing in a flower. Separately it will develop androecium and gynoecium. So the fertilization will take place, and those are the essential whorls of a flower. So in the flower, androecium and gynoecium differentiate and develop. So a typical flower has two parts. Which are the two parts? Androecium and gynoecium. In this, androecium is the male reproductive part and gynoecium is the female reproductive part. In this diagram, you can see here sepal is present, petal is present, then androecium and gynoecium. Gynoecium is present. Here the sepal and petal, they are the non-essential whorls, and the androecium and gynoecium, they are the essential whorls of a flower. Here you can see the structure of androecium. Here this is the structure of androecium and this is the gynoecium. Here in gynoecium you can see stigma, style and ovary. These together is called as stigma, style and ovary. It is together called as gynoecium. And androecium is the male structure in this, that filament and anther. These together called as androecium. Filament and anther together it is called as androecium. Androecium is the male reproductive organ. Now let's learn about androecium and gynoecium in detail. So first we are going to discuss about androecium. You can see in this flower, you can see the anther and the filament. These together it is called as androecium. Here you can see the anther and here this is the filament. This is structure of androecium. Androecium is the male reproductive whorl or reproductive organ. It is a male reproductive organ, and in this male reproductive unit, in androecium, that is a male reproductive organ. Which is the male reproductive unit in this? In this, the male reproductive unit is called as stamen. Okay. So the stamen of a flower, it is collectively called as androecium. The stamen of a flower, it is collectively called as androecium. Or you can see this is androecium. This group of stamen, it is called as androecium. So the male reproductive unit is called as stamen. Which is the male reproductive unit? Male reproductive unit is stamen. And stamen is also called as microsporophyll. It's very important MCQ, microsporophyll. Stamen is the microsporophyll, which is very important MCQ for NEET exam. You will get this question. Microsporophyll. Which part is known as microsporophyll? The stamen is known as microsporophyll. And in each species of plant, the number of stamens and the length of stamen, that varies. The number of stamen and length of stamen is variable in each species. Okay. So androecium is the male reproductive organ. The male reproductive organ is called as androecium, and the male reproductive unit is called as stamen. Here you can see the filament and the anther, that is together, it is called as Androecium. Androecium is a group of stamen. Filament and anther is called as stamen. Stamen means the filament and the anther. And the group of stamen, here you can see the complete group, it is called as Androecium, and the stamen is also called as microsporophyll. Microsporophyll. Stamen is also called as microsporophyll, very important, and the number and length of stamen varies in each species. Number of stamen and length of stamen, that varies in each species. Now let's learn the part of a stamen. Parts, which are all the parts of a stamen. The first is, here you can see, this is a stamen. Stamen, the filament and the anther together is called as a stamen. If you are cutting this anther, you can see this type of structure inside. These are the pollen sacs. So the structure of stamen, you will get a question for board exam to write the structure of stamen. So the stamen, in stamen, anther, the top part is called as anther and the down part is the filament. Okay, so in the picture here you can see two parts for a typical stamen. First is filament. Filament is long, slender, stalk-like structure. Here you can see it is a long structure and slender. It is a stalk-like structure. Okay, and on this stalk, terminally, here terminally you can see a structure that is bilobed, that is called as anther. On this filament, a bilobed structure you can see, that is the anther. This is the anther, you can see here, anther. So the parts of a stamen, which are all the parts of a stamen? Filament and the anther, you can see here. The long, slender, stalk-like structure, that is a filament, and anther. At the terminal position of the filament, you can see a structure which is bilobed, that is called as anther. Got it? And next, at the proximal end of the filament, or here you can see that proximal end of the filament, it is attached to the thalamus. That filament here in this picture you can see. See, this is a filament and the proximal end, the terminal end, there is an anther is present at the terminal end, and the proximal end, it is attached to the thalamus. Here you can see the thalamus, it is attached to the thalamus or the petal of a flower. In this picture you can see very clearly the proximal end of the filament is attached to thalamus or the petal. And the filament is long and slender. We have already told that filament is long and slender, and it is having a bilobed structure called anther at the terminal end. At the terminal end, the filament is having a bilobed structure that is called as anther, and at the proximal end, it is attached to the thalamus or the petal. So in the next picture, you can see the section of anther. Here in this picture, you can see the section of anther. And in the anther, you can see pollen sacs. These are the pollen sacs. See, here we have marked as pollen sacs. These sacs are called as pollen sacs, and inside the pollen sac, you can see the pollen grains also. These are the pollen grains. You can see the pollen grains also inside the anther. So the parts of stamen, filament and the anther. And the filament is long and slender stalk-like structure. Long, slender stalk-like structure, and the proximal end anther is, the terminal end anther is attached, and at the proximal end the filament, it is attached to the thalamus or the petal. It is attached either to thalamus or the petal. And in a typical angiosperm, the anther is bilobed. Here you can see bilobed. Two lobes are present in the anther. In a typical angiosperm, the anther is bilobed. And each lobe is having two theca. Two thecas are present in each lobe. Here you can see anther is bilobed. This is one lobe and this is another lobe. This is the first lobe and this is the second lobe. But each lobe, in each lobe, it is having two theca. See, this is the first theca and this is the second theca. Okay, in each lobe, in this lobe or in the first lobe, it is having two theca. Top one theca and down to second theca. Here also, it is having two theca, that is the enveloping sheath or case. In each lobe it is having two theca. The lateral that means the envelope or the sheath or the case. So in each lobe it is having two theca, or they are called as dithecous. They are called as dithecous. In the second picture you can see they are the, in anther it is bilobed and each lobe is dithecous. It is having two theca, and there are longitudinal groove that runs lengthwise to separate the theca. So to separate this theca, there are longitudinal grooves that runs. These longitudinal grooves runs lengthwise. Lengthwise they are running, and that separates the theca. That separates the theca. Now let's learn the transverse section of a young anther. If you are cutting an anther in a transverse direction or in a transverse way, we can see these type of structures. Okay. So I said stamen is having filament and anther. Right. So then if you are cutting and taking the structure, a transverse section in this anther, we can see the structure. We can see like this, the structure of anther. So anther, it is a tetragonal structure. Tetragonal structure. Anther is having a tetragonal structure, or it is having four sides. You can see four sides. For 1, 2, 3, 4. See this butterfly shaped structure. It is having four sides. First one, second one, third one, and fourth one. It is having four sides. So then in each side it is having microsporangia. Right? It is having microsporangia. In each side it is having a microsporangia. You can see here 1, 2, 3, 4. 4 microsporangia in each corner. So there are 4 microsporangia at 4 corners. These are the microsporangia. At each corner it is having 1 microsporangia. In 2 lobes. This is 1 lobe and this is 1 lobe. So in 2 lobes it is having 4 microsporangia. In the left side lobe, this is the first lobe. Here you can see 2 microsporangia, and the opposite lobe also you can see 2 microsporangia. 1, 2, 3, 4. So altogether there are 4 microsporangia. Right? There are 4 microsporangia, and these microsporangia, they will develop and it will form pollen sacs. This microsporangia will develop and it will form pollen sacs. And this pollen sac, that is the sac filled with pollen grains. What is pollen sac? that the sac which is filled with pollen grains. So that pollen sacs that extends longitudinally and through the length of that anther, this pollen sacs are extending, right? These pollen sacs are filled with pollen grains. Okay. So the androecium anther is a tetragonal structure. You can see the tetragonal structure here, and the microsporangia is located at each corner. At each corner, microsporangia is located. You can see here 1, 2, 3, 4. So there are total 4 microsporangia in an anther. So in each lobe, left lobe and right lobe, each lobe you can see 2 microsporangia. This is the one and this is second one. In this left side lobe you can see 2 microsporangia, and right side lobe you can see another 2 microsporangias. So there are 4 microsporangia altogether. So this microsporangia will develop into pollen sac. You can see pollen sac, and these pollen sacs are filled with pollen grains. Pollen sacs are filled with pollen grains. So the tetragonal structure, at the center it is having vascular strand, right? And here you can see inside microspore mother cell, and here at maturity, here the pollen is getting dehisced. So you can see inside the anther the pollen grains, numerous pollen grains will be present inside the anther. Then structure of microsporangium, right? So in this structure, in the first picture you can see a bilobed structure. Each lobe is having 2 microsporangia, right? 1, 2, 3, 4. 2 microsporangia. Here, this is an enlarged form. Here you can see anther. Anther is a bilobed structure, and each lobe you can see 2 microsporangia. In this lobe 2 microsporangia, here also 2 microsporangia, right? So we are studying one part of this microsporangia. We are going to enlarge one part. See, this microsporangia and this part we are going to enlarge. So you can see this kind of picture there, which is green color. If you are enlarging this part, we will get this type of structure. So if you are studying one part of this microsporangia, we can see that microsporangia, it is almost circular outline. It is having almost circular outline, right? And these microsporangium, it is surrounded by 4 wall layers. The microsporangium, here you can see the microsporangium, here the enlarged part, here it is surrounded by 4 wall layers. Which are the 4 wall layers? The first outermost layer is epidermis. Here you can see first outermost layer, that is epidermis, and second is endothecium. You can see endothecium just next to the epidermis. You can see endothecium, and third is middle layers. You can see middle layers. These 2 layers, they are called as middle layers. And last and innermost layer. Last and innermost layer is tapetum. So which are the 4 layers? Outermost layer is epidermis, then next to epidermis endothecium, then middle layers, and the innermost layer is tapetum. The epidermis, it is the outermost layer and it is a single layer. Epidermis is single layer. So this gives protection. That is the function of epidermis. What is the function of epidermis? To give protection. And now the second layer is endothecium. This endothecium, this layer helps in the dehiscence of anther or to release the pollen. So what is the role of endothecium? It is helping in the anther dehiscence or for the release of pollen. It is helping. And next is middle layers. Middle layers, these middle layers is having 1 to 3 layers of cells. Middle layers, it is almost 1 to 3 layered, and at maturity this layer degenerates. When the anther is getting matured, these middle layers, they degenerates, and these middle layers also helping for dehiscence of anther. It is also involved in the dehiscence of anther. Okay, so the structure of microsporangium, the microsporangium, it is nearly circular in outline, almost it is having a circular outline, and the microsporangium is surrounded by 4 wall layers. Which are the 4 wall layers? Epidermis, endothecium, middle layers and tapetum. Epidermis, endothecium, middle layers and tapetum. Those are the 4 wall layers. Epidermis, you can see the outer single layer, that is called as epidermis. Then endothecium, just next to the epidermis, endothecium is present, and middle layers, it is 1 to 3 layers of cells, middle layer. And this is also helping for the dehiscence of anther. Then last layer is tapetum. Tapetum is the innermost layer. And epidermis, endothecium and middle layers, all these 3 layers help in anther dehiscence, and it is giving protection. Epidermis, endothecium and middle layers, it is helping for dehiscence and it is giving protection to the anther. And next is tapetum, the innermost layer. So the tapetum is the innermost layer of anther, and this surrounds the sporangium, or this surrounds the sporogenous tissue. Okay, here you can see the tapetum. Tapetum is the innermost layer. In the previous slide, see, you can see it is the innermost layer. So we have talked about endodermis, epidermis, endothecium and middle layers. We have already discussed epidermis, protective, all these 3, epidermis, endothecium and middle layers, all these are having protective function and they are helping for the dehiscence of anther. So next is tapetum, the innermost layer. The tapetum is the innermost layer of anther, and this surrounds the sporogenous tissue. What surrounds the sporogenous tissue? Tapetum is surrounding the sporogenous tissue. Okay. These are the sporogenous tissue. The tapetum is surrounding the sporogenous tissue. And tapetal cells that gives the nourishment to the developing pollen grains. What is the function of tapetum? It gives nourishment to the developing pollen grains. And the tapetal cells having dense cytoplasm. Tapetal cells, they are having dense cytoplasm and they have more than one nuclei. Sometimes they have more than one nuclei also. Tapetal cells are having more than one nuclei. Important MCQ. They have more than one nuclei and they have dense cytoplasm, and their function is nutritional function. They are providing nutrition to the developing pollen grains, and they are generally polyploid. Generally, tapetal cells are polyploid. The tapetal cells that will show increase in the DNA content because they have more than one nucleus. So obviously they will show more than, or DNA content will be more in tapetal cells. In the picture, you can see sporogenous tissue, right? Sporogenous tissue you can see. Here, these are the sporogenous tissue inside this. So in young anther, this microsporangium, each microsporangium you can see sporogenous tissue at the center. In a young anther, at the center you can see sporogenous tissue, and these are diploid cells, and these are completely arranged, or they have compact, very compact arrangement. Which one? The sporogenous tissue, they are compactly arranged cells and they are diploid cells. Got it? And tapetum is the innermost layer. Here you can see next to middle layers, you can see the tapetal cells, and this is the layer which is giving nourishment to the developing pollen grains. Okay, that is a function of tapetum, giving nourishment to the developing pollen grains. And cells of this tapetum, tapetal layer, they have dense cytoplasm and they have more than one nuclei, and their function is providing nutrition to the developing pollen grains. And the microsporangium has sporogenous tissue at the center. At the center of this microsporangium, you can see sporogenous tissue, and this consists of homogeneous cells. The sporogenous tissue, they consist of homogeneous cells which are compactly arranged. These sporogenous cells are homogeneous cells and they are compactly arranged, and these cells are diploid. Sporogenous cell tissue cells are diploid cells. Got it? Next is microsporogenesis. Microsporogenesis. So from the title itself you can make out it is the genesis or the formation of microspores. This formation of microspores, it is called as microsporogenesis. So during the development of anther, each sporogenous cell, that means each cell of that center thick cells, okay, they are called as pollen mother cell. The center thick cells, they are called as pollen mother cell. Each cell of that, it is called as pollen mother cell. Why it is called as pollen mother cell? Because from those cells pollen grains are producing. So they are called as the mother cells of pollen grains. So these cells undergo meiotic division. They will undergo meiotic division. That sporogenesis, it will undergo meiotic division and it will form microspore tetrads. It will form microspore tetrads. You can see here microsporogenesis. It will undergo this sporogenous cells. It will undergo meiotic division and it will form microspore tetrads. So the formation of pollen grain from the pollen mother cell by meiosis, it is called as microsporogenesis. Formation of pollen grains. Pollen grains are getting produced from the pollen mother cell. From the pollen mother cell by meiosis. Their meiosis is taking place, and from pollen mother cell the formation of pollen grains, it is taking place, and this is called as microsporogenesis. And these tetrads, they separate and forms pollen grains. Here you can see the tetrads, and that tetrads will get separated and it will form the pollen grains, right? So microsporogenesis, as the anther develops, the cells of the sporogenous tissue, means at the center sporogenous tissues will be present in an anther, right? That tissue that will undergo, the cells of this tissue, sporogenous tissue, will undergo meiotic division. It will undergo meiosis, and after meiosis that microspore tetrads are getting formed. Here you can see microspore tetrads are formed as a result of meiosis, and each cell of the sporogenous tissue, each cell of the sporogenous tissue can give rise to microspore tetrad. So not only one cell, almost all cells, each and every cell of the sporogenous tissue can give rise to microspore tetrad. And the process of formation of microspore, the formation of microspore from a pollen mother cell or PMC through meiosis. So from the pollen mother cell, microspores are getting formed through meiosis. It is called as microsporogenesis. It is the genesis or the formation of microspore through meiosis. From what? From which cell it is forming? From the pollen mother cell. Which are the pollen mother cell? Each cell of the sporogenous tissue. It will act as a pollen mother cell, and through meiosis it will produce microspore. That is called as microsporogenesis. Next, when that anther matures, that tetrads, that dehydrates. and a microspore in that tetrad form. Here you can see after meiosis tetrads are formed. So when the anther is becoming mature, that dehydrates, that anther is getting dehydrated, or the water content of that anther will lose slowly as the anther matures. So the microspore in tetrad form, that also dissociate and develop into pollen grains. Here also the tetrad, that also will get dissociated and it will form the pollen grains, and each will form a pollen grain. So inside each microsporangium, thousands of microspores or pollen grains are formed. You can see thousands of microspores or pollen grains inside a microsporangium. So during anther dehiscence, these pollen grains are getting released. So all these pollen grains are getting released during anther dehiscence. So when an anther matures, that is getting dehydrated, and the microspore tetrads which are formed, that also dissociate and it will develop into pollen grains. So inside each microsporangium, thousands of microspores or pollen grains are formed. So during anther dehiscence, these pollen grains, they are getting released. Got it? See this tetrad, that also dehydrates and it will separate and form the pollen grains. Here you can see the mature pollen grains, and these during anther dehiscence, these pollen grains are getting released. Got it? It's clear? So that's all we have learned up to microsporogenesis. So the remaining things, now the pollen grains are formed. We have to learn about the pollen grain and all. We'll learn in the next class. So that's all. Thank you.